Thank you, Jerry. And didn't even fall through the trap door. That's great. Okay, so it's my great pleasure to call up the last speaker of the session, Professor David Langenau from Dana-Farber Cancer Institute, who will be discussing modeling p53 deficient cancers using zebrafish. So come teach us about zebrafish.
Great, thank you very much for having me. So I'm one of these unique people that was drawn in by a casual conversation with David Malkin about some unpublished work. And he said this would be a fantastic venue for you to come and learn about Li-Fraumeni syndrome. So thank you for having me. I'm learning quite a bit through the process. So I am Dave Langenau. I'm actually from Mass General Hospital. I trained at Dana-Farber and associated through the Cancer Center there. My laboratory is interested in how it is that cancers eventually go on to relapse and progress. And so my lab has really taken the notion that not all cancer cells are created equal. And so in this scheme, you can see that there are two distinct cell types. The red cell type, which makes up most of the tumor, really is not proliferative, whereas these yellow cells shown here are the cells that can remake themselves and go on to form and reform the cancers, which we call tumor propagating cells. And in fact, work in my laboratory has really focused on identifying genetic mutations that are acquired in these tumor propagating cells and assessing how they actually increase the overall self-renewal potential within these cells. So it's become appreciated that the same self-renewal programs that are found in stem cells are now recapitulated in these tumor propagating cells. And ultimately, our dream is that if we understand these processes, we'll be able to develop therapies that target those self-renewal processes and ultimately eradicate the tumors. So my laboratory is probably best known for using zebrafish as a cancer model. And in fact, the number one cancer that we study in my lab are sarcomas, and especially rhabdomyosarcomas. So the zebrafish is really a great tool because you can look inside the zebrafish. So for example, you can see here on the right-hand side, you can see individual cells that are labeled with different fluorescent proteins and dynamically visualize these in live animals. And importantly, on the left-hand side, you can actually see that zebrafish tumors are similar to what we see in human tumors, making our discoveries in the zebrafish really relevant to what we see in the human setting. So importantly, the zebrafish have been developed that generate a large different number of sarcomas, and in fact the ones in red have been developed in part by our group. We also now know that you can generate p53 deficient zebrafish, and actually these go on to develop a whole different set of tumors, which I'll talk about towards the end of my talk. So what I want to do is take you through how I got interested in p53 and Li-Fraumeni syndrome. So again, my laboratory uses zebrafish to study embryonal rhabdomyosarcoma, which is a common pediatric malignancy of muscle. Work from my lab has really established that RAS is the dominant oncogenic pathway in this tumor, which I'll talk about briefly. And we've known that p53 mutations are also fairly common in this tumor. And as you all know, Li-Fraumeni syndrome patients also go on to develop rhabdomyosarcoma commonly. And again, the biggest hurdle clinically for these kids is that they go on to eventually relapse, a large portion of them. And again, the tumor propagating cells are really the drivers of that process. So this is what the tumor model looks like. We've actually engineered the human form of KRAS into these fish. You can see that they have these sarcomas that develop immediately adjacent to normal musculature, shown on the right-hand side. You can actually see that these tumors are very invasive. In that histology in the intestine, you can see it completely effaces the intestine. And as I showed you previously, the zebrafish at the molecular level really seem to be most similar to human. Sadly, I don't have a pointer, but you can see that in the comparison on the left-hand side. Here we go. The human tumors compared to the zebrafish tumors here. So again, I think one of the major contributions that we had was nearly 8 years ago. We had actually used this molecular profiling to assess what pathways might be found in human tumors. And so, as I said, the zebrafish tumors really looked like human tumors comparing the embryonal versus the alveolar subtype. We also took this analysis and we looked at a whole slew of other types of tumors, and we immediately recognized that our zebrafish tumors really looked also like pancreatic adenocarcinomas, which, as many of you may know, have KRAS G12D mutations. And in fact, then we used bioinformatics approaches to look in a slew of cell lines that have been transduced with various oncogenes. And again, our gene signature was enriched with the RAS pathway. It took 7 years, but finally the St. Jude group and NCI actually sequenced these patients, and lo and behold, 90% of kids who have embryonal rhabdomyosarcoma, in fact, have RAS pathway activation as predicted by the zebrafish model. So using the model now, what we wanted to do was to try and isolate and understand those tumor propagating cells. So Myron Ignatius, a very talented postdoc in my group, had gone in and used fluorescent transgenics to probably make one of the most beautiful cancers that you'll see. And I'll show it to you in a minute. What he's actually been able to do is generate green fluorescent protein that is in the less differentiated tumor propagating cells. And as they differentiate, they'll turn on a red fluorescent protein. And as they terminally differentiate, they'll turn on a blue fluorescent protein. So what you can see in these pictures is a zebrafish with a tumor that's developed in its skeletal muscle. You can put these in a single drop of agarose and turn on a high-power confocal microscope and look inside them. And one of the initial findings of our work was that, in fact, these cancer stem cells or tumor propagating cells lie in unique niches compared to the more differentiated cells. And in fact, we saw the same thing when we looked at human patient samples. I'm in a pathology department, and in fact, one of the diagnostic tools they use is they say, well, patients who come in with embryonal rhabdomyosarcoma seem to have these patchy myoblasty type areas within the tumor, which had gone unappreciated, that likely indicates that in fact there are niches also similar to what we see in the zebrafish. So the other power of the approach is that you can actually leave these fish alive under this high-power confocal microscope and just watch what happens. So this is a 16-hour time-lapse video where you can see that the green fluorescent protein, again, which is the, sorry, I'll go backwards here. The green fluorescent protein is the tumor propagating cell. And again, they don't move very much. They stay in this metastable niche contained within the tumor. But now if you focus in on those red cells, they seem to be moving all over within the tumor. And in fact, as they terminally differentiate, those blue fluorescent cells really just hang out and don't do very much. So this led us to a conundrum, because in the field, everybody had believed that it was these stem cells that would actually be driving metastatic growth. So Myron went in and he actually imaged fish, and what he realized immediately was that, in fact, you can see that there's these areas, again, where there's the tumor propagating cells, but in the adjacent tumor area, those red cells are leading the charge and actually moving outside of the tumor. And in fact, if you come back days later, you can actually see that the green cells have now infiltrated into new areas of tumor growth, and you can clearly see there's a red front of cells that are moving into the newly forming growth. And in fact, you can also see this, we've just flipped the colors. On the bottom, blue now is those sinister myogenin positive cells, and you can see that in fact those cells are outside the large portion of the tumor, and in fact can infiltrate into the vasculature. So putting it all together, what Myron was able to show, quite remarkably, I believe, is that there's a tumor cell heterogeneity and division of labor. And in fact, that stem cells will, although they drive continued tumor growth, need not be the metastatic cell type, which I think had gone underappreciated until this work. And in fact, it's these very sinister myogenin positive cells that are the metastatic cell type. And the other corollary of this work now was that, in fact, these blue cells didn't seem to be contributing very greatly to the tumors. And so this led us to the hypothesis that if we were able to bathe these zebrafish in drugs, could we actually get them to terminally differentiate into these blue fluorescent types of cells? And so what Eleanor Chen did in my group when she was there, she worked with Novartis to actually do a large-scale chemical screen where she looked at 40,000 compounds that actually differentiated human rhabdomyosarcoma cells. She took the top 100 hits and then put them into a zebrafish cancer model, asking the simple question, well, does it actually affect overall growth? And then if it did it kill off those tumor-propagating cells? And in fact, this was a beautiful screen where she actually identified many drugs, several of which now, including the HDAC inhibitors, have really become center stage in thinking about clinical utility in rhabdomyosarcoma. So the reason I'm showing you all of this is to really give you a sense of the types of things that you can do and use the zebrafish models for, setting up what I think we can do in our Li-Fraumeni syndrome zebrafish model. I'll skip that slide. So we're not unique here. Tom Look and Len Zahn had actually made a Li-Fraumeni zebrafish in 2005, where they actually used an approach where they mutated the fish and then screened for them. They could see that these fish nicely went on to develop tumors, and in fact, they only develop, or largely develop, a single type of tumor, which is the malignant peripheral nerve sheath tumors, shown here. The only other type of tumor that they developed was a single melanoma. And so, although these fish have been incredibly useful for our field, there are some limitations. One of which is that the tumors develop over a long latency. This tumor spectrum is really restricted to these malignant peripheral nerve sheath tumors. And in fact, all those fluorescent transgenic approaches I showed you have not been, or largely not been utilized in these p53 deficient models. And then finally, one of the biggest hurdles is doing large-scale transplantation biology. I kind of breezed through this, but that's exactly what we did in our screen, where we took large numbers of fish and actually injected them with tumors in order for them to go on to get the same genetically defined tumor in a large cohort of animals. So again, Myron Ignatius, along with Madeline Hayes and my group, have now gone on and actually developed a full deletion of p53. And we can talk about why they did this at the end, rather than making patient-specific point mutations, for example. But what they did is they made these mutants, and you could see that, in fact, they completely lack p53, and in fact, they are born in Mendelian ratios. And remarkably, these fish go on to develop a very different spectrum of tumors. This may reflect that they're in a different genetic background than the p53 deficient tumors, or p53 mutant tumors from Tom Look's group. These are actually generated in genetically identical zebrafish, which, as we heard, can be very useful in sort of parsing out the randomness of what p53 deficiency eventually leads to. But what we can see is that there are distinct tumors that come up. We have malignant peripheral nerve sheath tumors, angiosarcomas, undifferentiated sarcomas, leukemias, and also, not on this slide, germ cell tumors. And in fact, we can see that just like in patients, there's a large spectrum, and we've looked at several of these, and more are coming. So we now knew that we had a pretty reasonable model, but could it actually be used for cell transplantation? And so the first types of tumors that were looked at were unlabeled, non-fluorescent labeled tumors. Again, these are in syngeneic backgrounds. So what Myron was able to do is actually, it's hard to appreciate, but this fish actually has a tumor near its eye, which is usually indicative of a malignant peripheral nerve sheath tumor. He was able to pluck out one of the eyes, it was a bilateral tumor. He could section it and actually see that, in fact, this looked like a malignant peripheral nerve sheath tumor. He was able to grind up the other portion of the tumor and inject it orthotopically or by intraperitoneal injection into the fish and recapitulate the same tumor. So the other types of tumors that we're developing, air quotes here, myeloid leukemia, we still have to work this up in any great detail, but these fish go on to develop these very robust phenotypes where you can see that the fish have these gill phenotypes and scale phenotypes. We now have innovated and actually now made the fish completely green fluorescently labeled, where that allows us to take these fish, isolate their marrow, and inject it into recipient animals. And what you can appreciate is that they robustly engraft into these fish. And again, now we can go in and we can use fluorescence activated cell sorting to isolate these populations, and lo and behold, they look a little bit like these myeloid precursor types of tumors. So more to be done here, but I think that we have a nice model developing of different types of leukemias. So of course, my lab is most interested in rhabdomyosarcoma, and so Myron was able to make rhabdomyosarcomas that were either wild type for P53 or had the mutation of P53, and then he could transplant these just as I had shown you. But as was nicely discussed earlier by Pierre, there's this concept in the field that in fact P53 may be expanding the stem cell compartment, or at least the cell types that initiate the tumor. But there's also a concept that P53 may be a self-renewal gene and cause them to continue to grow within the animals. So Myron was able to test this. This is a very busy slide. I won't take you through all the data, but the answer is when you put P53, when you lose P53 in this model, you actually have no effect on the overall frequency of these tumor propagating cells. Moreover, when we put this into a T-cell leukemia model, we see the exact same thing. So what we think is that P53 is really expanding the early population of cells that could be transformed, but once you become transformed, you really don't require P53 to modify the self-renewal effects that we see. The other real holy grail of using zebrafish is large-scale transplants to assess metastasis, which has been a huge hurdle in thinking about this in mice, for example. Although there are beautiful models, it is difficult to directly image this. So Chin Tang in my group has taken large numbers of animals where she can actually transplant them and then watch them grow and really say this tumor, for example, is not metastatic. Whereas this tumor, you can see at the site of injection, the tumor grows, but that it's metastatic and leads to tumor growth outside of the primary mass. And we can do this now in large scale. This is an animal study of 160 animals that have been transplanted, and it allows us to tease apart very subtle effects on metastasis, which I think you can appreciate in this graph. The red are the metastatic tumors, the blue are the infiltrative ones, and the green are really the ones that didn't metastasize at all. But this really uncovered a potent role for P53 loss in metastasis rather than in self-renewal. So I hope I've convinced you that the zebrafish is an interesting model and can be used in thinking about modeling Li-Fraumeni syndrome. I think one of the powers of what we've developed is that by having a complete lack of P53 in this model, we can actually go in now and take patient-specific mutations and engineer them into embryos and assess in functional assays how they might work. We can also model what happens in the setting of cancers, where you usually will have a point mutation that leads to a dominant negative and then loss of the other allele, which we now are able to mimic. I think this will allow us to develop new cancer models, as I showed you. My lab is also well known for doing evolution screens where we can serially passage tumors and follow how they genetically change over time and map those out using bioinformatics. I think that will be very useful. And then some other ideas that we have would be sort of thinking about chemical genetic screens, prevention screens. And of course, I'm new to this field, so any ideas you might have, please come find me. We're open to new collaborations and the like. So I hope I've convinced you the zebrafish is an interesting model. And with that, I'd like to thank my laboratory, including Myron, shown here, Madeline, and Chin Tang, who did a lion's share of the work, and all of our collaborators. Thank you.